Today we're looking at how lifespan is affected by pH limiting mitochondrial function in yeast. What are mitochondria? Mitochondria are where most of the cell's ATP is produced. It possesses its own DNA separate from the DNA of the cell. It creates its own ribosomes. It grows and divides on its own, unaffected by nuclear and cellular division.
First, let's talk about the relationship between vacuolar acidity and mitochondrial functionality. What makes vacuoles acidic is an enzyme called VATPase. As time goes on and cells reproduce and become mother cells, they decrease in acidity. In Hughes and Gottschling's study, they found that acidity consistently decreased in cells for the first 4 times they reproduced, but then became more stable and remained low for the following 18 rounds of reproduction. Now that we know more about the levels of acidity in cells over time, let's focus more on vacuolar acidity and how it affects the mitochondria. In this experiment, Hughes and Gottschling used an enzyme inhibitor called conc-A to block VATPase from working, thus lowering vacuolar acidity. In just 30 minutes, they saw mitochondrial depolarization followed by fragmentation and aggregation. Additionally, Hughes and Gottschling found that pH is an important predictor of lifespan, using conc-A again to inhibit VATPase in yeast cells. They found that these yeast cells had shorter lifespans.
Our next topic of discussion is overexpression of specific genes and their effect on dysfunction. In order to learn more about the mitochondrial function of the cell's genes were analyzed, and it was found that there are 250 genes associated with mitochondrial function in a cell. Then, every individual gene was modified to be overexpressed one at a time, and it was discovered that 2 genes, VMA1 and VPH2, reduced age-induced mitochondrial dysfunction significantly. In fact, dysfunction was suppressed in 93% and 77% of aged cells in which the VMA1 and VPH2 genes were overexpressed, respectively. VMA1 and VPH2 are 2 genes required for activity of the vacuolar H1 ATPase, which keeps acidity high in the vacuole. Therefore, it was then tested whether these genes reduced dysfunction by preventing vacuolar acidity decline, as the data would suggest. The results showed that, as expected, overexpressing VMA1 or VPH2 increased vacuolar acidity in at least 70% of aged mother cells. Although it is unclear how VMA1 overexpression increased vacuolar acidity, overexpressing VPH2 did so by increasing the levels of assembled VATPase at the vacuolar membrane. These results strongly suggest that reduced vacuolar acidity in mother cells leads to mitochondrial dysfunction. The next topic is regarding the rejuvenation of daughter cells even in aged mother cells. When analyzed, it was found that in new daughter cells, over 90% of vacuoles were acidic. However, in cells that had produced daughter cells, 87% had lost acidity, which highlights the progressive decline of vacuolar acidity with age. Given this information, it was of interest to determine whether new daughter cells' acidity content was a function of the acidity of the mother cell. Interestingly, after testing, it was concluded the vacuoles are regenerated with high acidity in newborn daughters and have no apparent relation to the acidity of the mother cell's vacuole at the time of reproduction.
Now let's discuss the importance of AVT1 and its effect on the vacuole and mitochondria. First off, AVT1 is a neutral amino acid transporter. To assess the effect that AVT1 would have on mitochondrial function, first Hughes and Gottschling used an enzyme inhibitor, conc-A, to block VATPase, thus lowering vacuolar acidity. They then overexpressed AVT1 and found that this disrupted mitochondrial dysfunction in 60% of cells. From this, they deduced that preventing proton-dependent neutral amino acid storage in the vacuole leads to mitochondrial dysfunction. To look more at the effect of AVT1, they went ahead and overexpressed AVT1 and found that it prevented changes in mitochondrial structure in 63% of older cells, even though vacuolar acidity was low in these cells. They then went ahead and removed AVT1 and found that it expedited age-induced mitochondrial dysfunction. Even when they overexpressed VMA1 and VPH2, without AVT1, mitochondrial dysfunction was not prevented. However, deletion of AVT1 did not increase vacuolar acidity. From these results, we can deduce that reduced vacuolar acidity in older cells might lead to mitochondrial dysfunction by preventing sufficient import of neutral amino acids through AVT1.
Now let's look at the effect of calorie restriction on lifespan extension. In yeast cells, calorie restriction uses at least 3 different nutrient sensing pathways, which are pKa, SCH9, and TOR. and so if any of these were to be reduced in their activity, it would mimic calorie restriction. When yeast cells were grown in calorie-restricted carbon sources such as 0.5% glucose, there seemed to be increased acidity in the vacuoles of young cells as well as relatively stable acidity in aging cells. This caused mitochondrial dysfunction to decrease in 90% of cells. As can be seen from graphs C and D, calorie restriction in wild-type yeast cells increased lifespan when no overexpression occurred. Conversely, calorie restriction did not seem to extend the lifespan of a cell overexpressing VMA1. And so from these findings, the researchers concluded that calorie restriction can ultimately lead to lifespan extension by preventing the decline of vacuolar acidity.
So why is this all important? Well, the discoveries made by Hughes and Gottschling through their experiments with yeast bring clarity to the events that comprise the aging process and pave the way to understanding how genetics and diet interact to influence lifespan, aging, and age-related diseases such as cancer and neurodegenerative disorders. In summary, the vacuole becomes less acidic relatively early in the yeast cell's lifespan, and that drop in acidity hinders the vacuole's ability to store certain nutrients. This disrupts the mitochondria's energy source, causing them to break down. Conversely, when Hughes prevented the drop in vacuolar acidity, the mitochondria's function and shape were preserved and the yeast cells lived longer. Hughes and Gottschling found that calorie restriction, limiting the raw material cells need, delays aging in part by boosting the acidity of the vacuole. This preliminary evidence in yeast of how calorie restriction extends lifespan can hopefully be translated to higher organisms like humans. In conclusion, given the similarities in the fundamental biology of yeast and human cells, the researchers' newly defined link between what cells eat and how they age could shed valuable light on the events that lead to age-related disorders in humans.
Thanks for watching!